Welcome back to Share Science, the weekly science brief for June 29th, 2026. I'm Curie.
And I'm Derek. This week, IBM unveiled the world's first sub-1 nanometer computer chip, cramming nearly 100 billion transistors onto a piece of silicon the size of a fingernail, which is wild. I mean, DNA is already microscopic, and now we're talking about technology operating at an even smaller scale. What does that even mean for the future of computing?
It's a complete game changer. This chip, built at the 0.7 nanometer node, packs nearly 100 billion transistors onto a piece of silicon the size of a fingernail. To put it in perspective, this is the first time we've gone below the 1 nanometer threshold, which is something people thought might not even be possible.
So, how did IBM manage to pull this off? I mean, we've been hearing for years that we're approaching the physical limits of how small we can make transistors.
That's exactly what makes this so impressive. Instead of trying to shrink transistors on a flat plane, IBM went vertical. They developed a new architecture called nanostack, where they stack and stagger transistors in 3 dimensions. Think of it like building a skyscraper instead of spreading out a single-story building. This allows them to pack far more transistors into the same footprint.
And the performance gains must be huge, right? I mean, we're talking about a leap forward in computing power.
Absolutely. Compared to their 2 nanometer chip from 2021, this new design delivers up to 50% higher performance or up to 70% better energy efficiency. That energy efficiency is especially critical right now because AI data centers are consuming staggering amounts of electricity. A chip that can do the same work with 70% less power is a massive step toward making AI more sustainable.
It's fascinating to think about how this could reshape industries. From AI to cloud computing, this kind of technology could have ripple effects everywhere.
Exactly. And while IBM doesn't manufacture chips at scale, they develop architectures and license them to companies like Samsung and Intel, they estimate we could see commercial adoption of this technology within 5 years. It's a glimpse into the future, and it's incredibly exciting.
All right, let's move from the microscopic to the medical. This week, scientists announced they've cracked an undruggable pancreatic cancer target. That's a term you don't hear every day. What does it mean?
It's a striking term, isn't it? In cancer biology, undruggable refers to proteins that are incredibly difficult to target with drugs. These proteins might lack obvious binding sites for drug molecules or be so essential to normal cell function that blocking them would be too toxic. Pancreatic cancer has one of these notoriously undruggable targets, and it's been a major roadblock for decades.
And pancreatic cancer is one of the deadliest cancers, isn't it? The survival rates are just devastatingly low.
They are. The 5-year survival rate is around 12 to 13%. It's often caught late, spreads aggressively, and has been resistant to the kinds of targeted therapies that have revolutionized treatment for other cancers. So any progress here is monumental.
So what did the researchers do? How did they finally crack this target?
The breakthrough involved a novel approach to engaging the target. The specifics are still being analyzed, but they found a way to disrupt its cancer-driving activity in a way that previous drug designs couldn't. In preclinical models, this nearly doubled survival rates. Now, it's important to note that this is still early-stage research, but it's a significant scientific milestone.
It's a reminder of how complex and challenging cancer research is, but also how perseverance can lead to breakthroughs that once seemed impossible.
Exactly. And it opens new doors for treatment strategies, not just for pancreatic cancer, but potentially for other cancers with similar challenges. It's a hopeful step forward.
Speaking of hope, let's talk about our next story, Earth potentially seeding Venus with life. This one feels like it's straight out of science fiction.
It does, doesn't it? But it's grounded in serious scientific modeling. The idea is that asteroid impacts on Earth could eject rock and microbial life into space, and some of that material could end up on Venus. It's a process called panspermia.
We've talked about panspermia before, usually in the context of Mars. Why Venus, and what makes this idea plausible?
Great question. Mars often gets the spotlight, but Venus has intrigued scientists for years, particularly its cloud layer, about 50 kilometers up. While the surface of Venus is hellish, hot enough to melt lead with crushing pressures, the cloud layer is much more temperate. The study modeled the trajectories and conditions for Earth to Venus transfer and found that it's physically plausible for microbes to survive the journey and potentially find a hospitable environment in those clouds.
Of course, this doesn't mean there's life on Venus. It's more about the possibility, right?
Exactly. This study doesn't provide evidence of life on Venus. It just adds to the scientific conversation about how life might travel between worlds. Upcoming missions to Venus will investigate its atmosphere, and if we ever find something there, we'll have to seriously consider whether it came from Earth.
It's fascinating to think about the interconnectedness of planets and the potential for life to spread across the cosmos.
It really is. And it's a reminder of how much we still have to learn about our own solar system.
All right, let's move on to something closer to home, literally. Researchers have discovered that neurons break their own DNA as part of brain development. That's not what you'd expect.
Not at all. DNA is usually something cells go to great lengths to protect. Double-strand breaks in DNA are associated with cancer, cell death, and aging. So, to find that neurons deliberately break their own DNA during development is a complete paradigm shift.
Why would they do that? What's the purpose?
As the brain develops, newborn neurons have to migrate to their final positions. This involves squeezing through incredibly tight spaces in the developing tissue. The mechanical stress of this journey causes the DNA to break. But rather than being a catastrophic accident, it's a managed part of the process. The neurons have repair mechanisms ready to fix the damage once they've reached their destination.
It's like the cells are saying, this is going to hurt, but it's necessary.
Exactly. And it raises fascinating questions about how this process might influence brain function and development. Could errors in this repair process contribute to neurodevelopmental disorders? It's a whole new area of inquiry.
It's amazing how much we still have to learn about something as fundamental as brain development.
Absolutely. And speaking of fundamentals, let's wrap up with our final story: gum disease and a tropical fruit that might help reverse its damage.
Gum disease is one of those things people tend to overlook, but it's a serious health issue, isn't it?
It is. Periodontal disease is a chronic inflammatory condition that can lead to tooth loss and has been linked to heart disease, diabetes, and other systemic health issues. Current treatments can manage the disease, but rarely reverse the damage.
And this is where the tropical fruit comes in?
Yes, researchers found compounds in a large tropical fruit that appear to promote tissue regeneration in the gums. These compounds have anti-inflammatory and tissue regenerative properties, creating an environment where healing can occur. It's early research, but it's a promising direction for better treatments.
It's a reminder that solutions to big problems can come from unexpected places.
Exactly. And it's a hopeful note to end on. From the tiniest chip to the vastness of space, science continues to surprise and inspire.
It really does. Thanks for the conversation today.
Thank you. And to everyone listening, stay curious. Until next time.